Okay, good afternoon to everybody. First of all, I would like to join the other speakers to thank the organizing committee to give me the opportunity to join some of our results today with you. This is an ongoing project related to protein alteration in relation with skeletal muscle aging. Just a quick overview. As has been said yesterday in the afternoon session, a common feature of the aging process along different tissues and organs is the dysregulation of protein homeostasis. And one of the factors involved in this dysregulation is increased protein modification due to increased free radicals. In particular, I will focus during this talk in protein oxidation. And when I talk about protein oxidation, I'm referring particularly to protein carbonylation. So, as we hear from yesterday from Dr. Malcolm Jackson, skeletal muscle aging is characterized by a decrease in skeletal muscle mass and number of fibers during aging. This results in a progressive loss of mobility and decrease of quality of life with important social and economic problems. I will focus mainly in a population of the skeletal muscle, which are adult muscle stem cells, also known as satellite cells or myoblasts. These cells are located beneath the basal lamina and are very important for skeletal muscle regeneration. After injury or damage, these cells replicate as myoblasts and they differentiate to form new myofibers. Importantly, during the aging process, these cells have been shown to have a decreased ability to proliferate and differentiate, and in somehow explaining, at least in part, the decreased muscle mass observed. Previous studies show that under acute oxidative stress, these cells has a limited, a reduced proliferative capacity. Here you can see that only around 30% of the cells were able to include BRDU, so suggesting a stress-induced premature senescence phenotype. Also, senescent cells has a reduced ability to form myofibers, and you can see here that only a few percentage can form more than 50 myonucleases per fiber. So this is our model. We obtain cells from young donors, muscle biopsies. We purify myoblasts. Here you can see the cells at population W30, and during replicative senescence, when they reach senescence at population W around 48, they show the clear phenotype of senescent cells, changing morphology, and also they show increase in P16 levels, which is related to cell cycle arrest. So we then look at the peptidase activities of the proteasome. As we hear from yesterday, it has three main, the 20S has three main catalytic activities, the chymotrypsin-like, trypsin-like, and caspase-like. We observe a decrease in the three peptidase activities during replicative senescence. Here in the x-axis you have population W30 until senescence, the same for the trypsin-like and caspase-like. Most interesting, we didn't find a decrease, at least of the catalytic subunit. Here you have beta 1, beta 2, and beta 5, and the levels of protein didn't change from young to senescent cells, suggesting the presence of internal inhibitors of the protein. Then we look at carbonylated proteins, again in the same model, replicative senescence of this human myoblast. Since carbonyl group do not have a distinct signature, we use a typical approach of derivatization of the carbonyl group with nitrophenic hydrazine to form nitrophenic hydrazone, following immunodetection with antibodies to the DMP product. What we observe is an increase in protein carbonyls during replicative senescence. Then we move forward to the identification to which are the targets of carbonyl modification. This is the technical rationale. We went first to separation by 2D-dimensional electrophoresis of the proteome, followed by 2D Western blot for immunodetection of carbonyl groups, following identification of the selected spot by MS. Here is how the oxyblot looks like. We observe clearly an increase in the number of spots on senescent cells when comparing to young cells. Then we moved to the identification. We found that most of the proteins being carbonylated belong from the cytosolic compartment. However, also proteins from the nucleus, membrane protein, as well as mitochondria has been identified. When we group these proteins in terms of functional characteristics, we observe key cellular pathways involved, such as carbohydrate metabolism, cell morphology, and so on. Importantly, when we group these proteins identified, here you can see in red in a pathway analysis, we observe that they have interrelationship between each other, suggesting that protein carbonylation is not a stochastic process. Importantly, we found interesting proteins such as TNF-alpha, HSP70, and Huntingtin in some of their nodes. So then we move to another model. This is the same as human myoplasm, but now under acute oxidative stress. Briefly, what we use just an stress with hydrogen peroxide for 1 hour, using 2 concentrations, and then we perform a parallel proteomic approach. One looking at changes at the proteins at the expression levels using the 2D DICE technology, and also looking at the oxyproteome. Here are some representative cells of the 2D DICE approach. Here you have the control cells that were labeled with well-fluorescent tag, proteins from oxidized cells with the other. We perform principal component analysis, and we were able to separate the two groups in the analysis, validating this approach for proteomic analysis. These are the proteins that we identify whose protein whose levels were changed upon acute oxidative stress. We found proteins involving antioxidant response, of course, such as peroxyroxine 6 and protein DJ1. We found proteins involving energetic metabolism, protein folding, and protein degradation. So then we performed the oxyproteome of these cells under acute oxidative stress. Again, to the oxyblot, we observed only a restricted set of proteins that belong carbonylated. These are the identification of the proteins. Again, we observe proteins modified in very important cellular pathways, such as protein synthesis, elongation factor 2. We also found proteins involving energetic metabolism. And well, when we perform cellular pathway analysis, here is a pathway analysis for the carbonylated proteins, and here for the proteins differentially expressed. We didn't find at all overlapping of the pathway resulted. Here we have Huntingtin in the central node for the carbonylated protein, and we have TP53 for the differentially expressed protein, underscoring the importance to do proteomic analysis looking at the expression level, but also at the modification level. However, when we look at the proteins that we identify carbonylated in 2 approaches, we found almost a 45% of overlapping, suggesting that there is a susceptibility of some of the proteins to become modified. And when we look at the pathway analysis, we observe proteins in common for the acute oxidative stress and for the senescent cells, such as Huntingtin, and also we observe TNF-alpha and MYC as different nodes. Finally, I would like to thank the people in the lab, principally Professor Bertrand Figuet, who gave me the opportunity to join his lab, Janek Gisevich, who performed basically the 2D oxyblot, Roman Laduz, who I collaborate for the 2D DICE, then the people in Denmark, Peter Rovstov and Elina Rowoska, who performed the identification for mass spectrometry, and people here in Paris in the Neurology Institute, Vincent Mounier and Juliette Butler-Brown. Thank you very much.
The paper is open for questions, discussion from the audience. Okay. Your central hypothesis was that H2O stress and senescence would activate a similar number of proteins at oxidation, and you concluded that 45% of the proteins had an overlap. Right?
Yes.
My particular interest is in peroxyredoxin 1 and 6. Could you comment a bit further about that?
Yeah, we found, this is a very interesting point, since In the Dutch experiment, I mean, when we look at the different expression level, we found changes in peroxyredoxin 1 and 6, but we didn't identify them carbonylate. However, it has been described other type of modification for peroxyredoxin 1 and 6.
So in your study, no carbonylation was...
No carbonylation of peroxyredoxin 1 and 6. But it has been reported other type of oxidation.
Any reason why you think you didn't pick that up in your cell type?
That's a good question. The issue is that, I mean, the approaches are also different. No? The Dutch approach is a little bit different from for immunodetection of carbon. It's probably, I think that if we can go to more sensitivity approaches for detect protein carbonylation, we can.
And in terms of the transcription factors, I think you had elongation factor, and I couldn't pick up the rest. Are there any redox signaling factors, transcription factors that are altered?
Yeah, actually we found MYC, which is quite interesting. Yes, but for sure, I mean, I think that the important aspect of the study is to underscore the dual approach of looking at proteins that change in the expression level, but also the proteins that are modified, and show that actually they are not much overlapping.
Any other questions? The audience, please.
I have a question about the analysis. When you show an overlap from senescence versus a stress, what statistical caution do you take to avoid that being a random event?
Well, actually, what we did is to perform the same study different times. I mean, with different batch of cells, and actually, what we found is consistently the same spot modified. At the level of statistical analysis, I mean, we haven't done much at this point. What is interesting to us at this point is that probably suggests that there is a role in oxidative stress in the development of the senescent phenotype.
I guess the fundamental question is how do you sort of validate that the overlapping proteins?
Are the proteins that belong from the two different studies?
Yeah. Okay.
Mainly are more, I mean, found in.
Martin, the difference between the senescence approach and the oxidative stress approach is that in oxidative stress you incubate the cells with hydrogen peroxide. Now, in terms of senescence, could you measure the levels of hydrogen peroxide in the senescent cells to see whether actually one model could be compared with the other?
Yes, actually that will be something very interesting to do. We haven't done that, but probably yes. I mean, what is also an interesting question is which is the source of reactive oxygen species that give rise to the senescent phenotype at the protein oxidation in senescent cells.
Well, that comes back to Giovanni's question about the peroxyredoxin 1 and 6, because there should be a mechanism that sort of universal for the inactivation of those proteins.
Sure.
And it's very likely that in the senescent model you might have certain levels of H2O2 that are required for the identification of those two proteins there, which basically are resistant residues which are sensitive.
Yeah, sure. No, that has been described also. Yes, actually we identify peroxyredoxin 1 and 6 only in the acute oxidative stress. We haven't identified in the Dutch analysis of the senescent cells that I didn't show today. But makes sense, I mean, because it's probably the first line of defense to peroxide.
Thank you very much. Next speaker.